Hey, Leanne, thank you very much. It's a great pleasure to be here. I appreciate being invited to participate. Uh, the integrated pest management approach has been a part of mosquito control long before it was a recognized practice. As early as the early 1800s, there was an area by Savannah, Georgia that switched from wet rice culture to dry rice culture and used local tax monies to do it. So this is kind of an unrecognized mosquito control event. The earliest ditching of the New Jersey salt marshes and the open marsh water management practices that take place today are other good examples of an integrated pest management approach. I grew up on the salt marshes of New Jersey and Ocean County, very familiar with this area and the ditching of the marshes and the effectiveness of the salt marsh mosquito and floodwater abatement there. Before pesticides, this is the way they did mosquito control. So today, we're returning to a more integrated approach, and we're going to hear a lot about the different, some of the approaches used for this. There's many reasons to return to an integrated approach and use an integrated approach. The efficiency and the effectiveness of the control, the sustainability, and public perception. I think we all know that the public's probably going to have more input into our mosquito control practices moving forward than they have in the past. Integrated mosquito management is a kind of a broad topic that I have a little bit different approach to than some people. I serve as the public health extension specialist here in Georgia, and we have many very small programs where there's limited resources and the populace only wants certain aspects of mosquito control done. So while that may not be a comprehensive program, we do have comprehensive programs. Chatham County down in Savannah, as many people are aware, is a great program, uses all facets. But some areas want to just use education, and this involves educating the public as to what they can do around their homes. And I also include educating the workers. We've got, almost 500 people here today. Hopefully everyone will take up, a couple tidbits of information they can take back to their programs. Workers become more confident in their knowledge, and you can deal with the public better. And the more confidence you have when you're dealing with the public, the better it represents the program, the better the public thinks of our programs. Source reduction will be the next step. Permanently eliminate sites, improve drainage and habitat manipulation, the marsh management and flooding practices that occur in the lagoons in Florida, but community cleanups as well. So source reduction is and we can do that without even knowing what mosquitoes are there, because we all know that a roadside ditch that holds water is a problem. Surveillance would be the next step. What do we have and where do they occur? This helps focus our efforts. As with all pest control, it's obviously best to know what species you're dealing with. For mosquito control, it's more important because it tells us where to look as far as the larval habitats. Larviciding would be the next step. We target the habitats that can't be eliminated. Larviciding requires effort and resources. I think people sometimes overlook this. Those of us who have been involved in larviciding efforts know that it's a lot of work, requires boots on the ground and resources. And the final step would be adulticiding. Adulticiding is highly effective. Communication becomes essential. We need to be aware of pollinator concerns, and insecticide resistance is obviously a very important aspect moving forward here. And we do have a cooperative extension circular 1154 that goes into more detail on these. Larviciding has many advantages. The larvae are concentrated in a confined or defined area, particularly compared to the emerging adults. The larvae are essentially immobile, and they're not getting away for a few days, and they're accessible. The CIA approach is something that William Horsfall was a proponent of and talked about in his 1985 50th anniversary presentation at the AMCA meeting. So this goes back a ways. I'll add to that the advantages of larviciding is we typically think that the larvae are susceptible. Obviously, there's some resistance occurs, but in general, I think the larvae are more susceptible than some of the adult populations. It's proactive. Obviously, we're targeting the larval stage before the adults emerge. And it's more acceptable to the public, has reduced risk to pollinators. Choosing a larvicide is a really important decision, and there's going to be, I think we're going to have some good presentations about this moving forward. Obviously, there's a wide variety of formulations. We have 4 types of active ingredients for very different modes of action. And ideally, the formulation and active ingredient is chosen based on the type of habitat and the mosquito population that's present. And that's really what is the fine tune to which product is used, which formulation and active ingredient is the habitat and the stage of the population, how much time you have to get them before they get off and things like this. The more experience and knowledge that the operator or program has, the more targeted and efficient the larvicide application will be. The larvicide active ingredients, we basically have 4 groups. There are insect growth regulators, which are absorbed through the integument of the larval stage. S-methoprene is the most common and has been a long time standard. The Insecticide Resistance Action Committee, or IRAC classification, is a 7A. This describes the mode of action of the larvicide. Pyriproxyfen is also an insect growth regulator that's approved probably for storm drains. I don't think there's a floodwater formulation, so we're not going to talk about that anymore. Just want to touch base on it. The microbial products, these must be ingested, the proteins that are produced by the bacteria. Bacillus thuringiensis, subspecies israeliensis, and Bacillus sphaericus. Lincillibacillus is a new name. I think we've been through the nomenclature changes with mosquitoes, so this is just something to be aware of. I'm going to stick with the Bacillus sphaericus moving forward. Most people are still most familiar with that. Surface oils produce a physical barrier. Today they're highly refined mineral oils, and there's no IRAC classification because it's a physical control agent. Spinosad is effective by contact or ingestion, and this is a biological neurotoxin with the IRAC 5 classification. So we've got 3 different classifications in the physical barrier. So for larvicides, that's a pretty wide range of modes of action, and it's probably a little bit better, a little bit more advantageous than our adulticiding options. The insect growth regulator S-methoprene. This is a natural juvenile hormone that was first identified in 1967. It's involved in the regulation of the physiological processes, particularly metamorphosis, and research quickly followed, developed the first larvicide around 1975 for mosquitoes. It's used in a wide range of insecticides today. My first experience was with hornfly control on livestock. It inhibits ecdysone from initiating the molting processes. It's particularly disruptive to adult emergence, and malformed adults may be observed on the water surface of treated areas. The pupal stage does not feed, as we're all aware, so they will eventually deplete the body stores of essential nutrients and starve. So it primarily affects the pupal to adult transition stage, and it's effective against all mosquito species. Just an example of how it works: normal mosquito development, it would have high juvenile hormone levels in the early instar larvae. They decline as they age and mature and approach the pupal stage. When you treat a habitat with the juvenile hormone analog, methoprene, you maintain these high levels of juvenile hormone in the environment, and it just disturbs the physiological processes of the pupal stage primarily. So again, it's absorbed through the cuticle and by ingestion. The concentration must be higher in the larval environment than circulating in the larval body for disruption to occur. It's used for the wide range of applications. Larvae and pupae don't die immediately, so this leaves them in the food web. This has some advantages. Efficacy evaluations can be challenging because the larvae and pupae don't die. So that's something that operationally technicians need to realize, that you can have a very effective methoprene treatment ongoing and the larvae are still there. So that's something people need to be aware of. And it's a long time standard. There's been many improvements in the formulations. There's formulations available from several of our producers now. Microbial-based products, Bacillus thuringiensis, subspecies israeliensis, or BTI, is probably the most common. Again, the Bacillus sphaericus is also available. Both of these materials are very common today. BTI probably having a more prominent role in targeting floodwater mosquitoes. Bacillus sphaericus was more commonly recognized as a material used to target culex mosquitoes, particularly for the reduction of West Nile virus transmission, but the other uses are expanding. It's very effective against the Serophora mosquitoes that Barry just talked about, and also Aedes vexans. So it definitely has uses in the floodwater realm. And there are combined formulations available, both Sphericus and Israeliensis, that will provide a wider range of treatment options in a wider treatment zone. Bacillus thuringiensis israeliensis is a bacterium that occurs naturally in soils and aquatic environments worldwide. BTI was discovered in a drying pool in a riverbed in the Negev Desert of Israel in 1976, was quickly produced for commercial products. Thuringiensis had long been recognized for having insecticidal properties. There's other BT products for lepidopteran, forestry products, but the discovery of BTI brought it into the public health arena. There are many serotypes and strains of the BTI bacteria, and the different producers use different strains for their actual final products. So that's something you'll see sometimes on the label. The active ingredient of BTI-based formulations are delta endotoxins that are produced at the time of sporulation. The endotoxins are composed of 4 proteins that are activated by the proteolytic gut enzymes in the alkaline gut environment of the Nematosser and Diptera larvae. All right, a lot right there. Nematosser and Diptera larvae are your mosquitoes, black flies, and Chironoma midges. They have a very high pH in their midgut, which is unusual for most animals and organisms. And they have the enzymes associated with that highly alkaline gut are effective against these proteins, cleave them and activate them. Just an example of that, the image there, the midgut of a mosquito larvae. The formulation is the protoxins are ingested. They're activated by the enzymes, break them down. The polypeptide fractions, or which are proteins, bind to the cells. They form pores or holes in the lining of the gut cells. An osmotic imbalance occurs. The cells swell, they lyse, the larvae die. This can happen rather rapidly when they're exposed to high concentrations. BTI must be ingested to be effective. Larval feeding is absolutely critical. And the larvae seem to be very susceptible to these proteins in this product. It kills the larvae very quickly that are exposed to high concentrations, slower for those larvae exposed to lower concentrations. And this is something that's operationally, people need to be aware of that, just because low populations killed with, treated with high concentrations may die very quickly, but larvae exposed to lower concentrations still probably will experience mortality. It might just take longer time. Appears to be the amount of protein they ingest, how rapidly that midgut is disrupted. As I mentioned, there are 4 proteins associated with larvicidal activity, and there seems to be some synergistic effects of these proteins. They're cleaved by the enzymes. You often see about a 25 kilodalton protein. Consequently, there seems to be a reduced risk of resistance with BTI. There's multiple proteins involved. It seems it's a very complicated process and very specific environment where it occurs. There are formulations of BTI that are approved by the Organic Material Review Institute, or OMRI, and that's a very good thing. Obviously, some of the formulations are very clean and can be used in organic production areas. Their BTI has no mammalian toxicity, and the non-target impacts are very minimal. Bacillus sphericus, another bacterium that occurs naturally in soils and aquatic environments worldwide. It was first isolated in 1964 from dead Culicida incidens larvae in California rock pools. There are several serotypes with larvicidal activity, but it has a narrower spectrum of activity, as there are only 2 proteins involved with the toxic effects of it. Has a similar mode of action to BTI, but that's kind of a general statement. It affects different cell structures than BTI, but it is ingested. The toxins are attached to a living spore, and the coating of the spore seems to aid in the residual aspects of the Bacillus sphericus larvicides. There are also is some recycling of toxicity associated with Sphericus. The greater the larval population in initial treatment, the greater the residual effect. So what's going on there, the larvae are actually ingesting spores, and these spores will germinate and produce bacteria in the larval cadaver to produce a recycling event with the Bacillus sphericus to extend the residual control up to around 28 days. It is also approved by the National Organic Program. Some of the formulations are, so it also has a very clean profile as far as environmental and organic production areas. Just a quick example of the mode of action. The spores in associated protoxin are ingested. Feeding ceases, the spore coat dissolves, releasing the protoxins. The protoxins or the proteins are cleaved by enzymes, activated, and bind to the gut wall, creating pores or holes in the gut wall. The cells lyse. The larvae die, but some recycling will occur because, as you recall, the spore is still there, and the cadaver environment of the cadaver is very conducive to bacterial growth. Surface oils, petroleum oils were the first chemicals used in efforts to control mosquito larvae. Thankfully, we have progressed to a more refined mineral oil-based product today. These are the only pupicides, so that's something to take out of this right here. It's an extremely important aspect. If you've got habitats that have large pupil concentrations, you've got limited options of what you're going to do. And also, they're coming soon. Adult mosquitoes are coming soon. So surface oils, the mineral oil-based products can be used to get these. The effectiveness of these oils are limited to mosquito larvae and pupae that breathe at the water. Breathe air at the water surface. So your Coquetidia, the mosquitoes that will insert their air siphon into the roots of plants are not going to be affected. Lower rates will minimize non-target impacts. Higher rates are required for polluted water in areas with significant vegetation. This material's got to spread across the surface of these areas, so application will be important as well. It's probably the least studied larvicide. I thought that was interesting. I was looking at some of the Florida information, and John Beadler stated that, and the mode of action is related to the effect of the product on the surface of the water, and there may be some toxic impacts as well. So it's interesting that the oldest material is maybe the least well understood. Suffocation of both the larvae and pupae does occur. This mode of action is completely different from the other larvicides, so it's very difficult to envision how resistance would occur to this material. Coverage is very important, and mortality can be rapid. Spinosad is a biological neurotoxin. It's one of our newest active ingredients. Has a kind of an interesting background story as well. A vacationing scientist took a soil sample from a drum that was used to make rum in the Caribbean. From that sample, a new species of bacteria was identified, Saccharopolyspora spinosa, and this translates to spiny sugar. So during the fermentation of the bacteria, the two most prominent and active compounds in the fermentation broth are spinosin A and spinosin D, hence the term spinosad. This material alters the function of the nicotinic and GABA-gated ion channels, causing rapid excitation of the insect nervous system, leading to involuntary muscle contractions, tremors, paralysis, and death. So this is, kind of your traditional neurotoxin. You got the synapse there and the acetylcholine effects. So it's more of a traditional material and different from our other active ingredients. This neurotoxin is specific to invertebrates, and it is classified as a reduced risk pesticide by the EPA. It has also been approved for use in organic productions areas by OMRI, so it also has a pretty clean environmental and label approach. It breaks down rapidly in sunlight and in soils, and the microbes are going to break down spiny sugars, as you would imagine. It's effective against all larval instars, mortality most rapid when ingested, but some contact toxicity. And it is a unique active ingredient whose use is growing and is certainly an excellent part of a rotation. So our larviciding options, we have many different formulations that are available. There are combinations of active ingredients that provide more application flexibility, a wider treatment window. That would be the BTI and methoprene combinations, or BTI and sphericus combinations. It's been going on for a long time. The combination formulations have probably been more refined now in more recent years with formulation technology. Matching the optimal formulation and active ingredient will maximize the efficiency and effectiveness of a larvicide application. That's really something I took out of this as I prepared for this presentation. When I was talking to many of our industry representatives, I highly suggest that we discuss the strengths and weaknesses of formulations with your commercial representatives and other control specialists. Use your resources. To any directors out there, as we return to trade shows after this pandemic, I strongly encourage the support of operational people participating in these meetings and developing a network that is so valuable. There's so much information out there. We can't learn it all ourselves, but our commercial representatives and our control specialists from adjoining counties and states can provide information and improve our programs tremendously. So use your resources. Networking is really important. Product rotation remains an important aspect. We've got a lot of different very good products here. Integrated mosquito management has never been more important. Using all aspects of it: education, source reduction, surveillance, larviciding, and adulticiding. Larviciding can be highly effective in preventing all types of mosquito populations, whether it be floodwater or containers. A lot of work been done in this area in recent years. Larval surveillance and knowledge of the physical parameters of the larval habitat is critical to choosing the proper product and optimizing the timing of the application. That's what we have all these different formulations for, because there's whether you have single brood, whether you have extended release, multiple broods, the timing, late instar, just a lot of different aspects that the various formulations can help us address, and the expertise of our commercial reps can help us choose which one. And of course, being cognizant of non-target impacts, I want to stress that we're always concerned about what else we're affecting. In summary, the wide variety of floodwater habitats can all be satisfactorily addressed by one or more of the active ingredients or combinations that are available. We have 4 very different modes of action: the insect growth regulator methoprene, the microbials and their stomach actions, and being Thuringiensis, Israeliensis, and Bacillus sphericus. The surface oils, highly refined mineral oils, again, our only pupicides, and the biological neurotoxin spinosad provides a very different mode of action from the other larvicides. Effective surveillance of the target population, site specificity, and cost will often dictate options. I am not so naive to think that cost does not come into play. I do want to stress that there are other costs involved, whether it be susceptibility or environmental impacts. So it's not just the dollar and cents of the product, but all the costs involved. Susceptibility is a big thing in product rotation. So cost averaging over 2 or 3 seasons might be a better way to approach the cost aspect moving forward. But all the active ingredients can and should play an important role in floodwater mosquito management programs moving forward. Lastly, I'd like to put a plug in for the Georgia Mosquito Control Association. We're going to meet in October, and we want you folks to come join us. Thank you.
Awesome. Thank you, Elmer. We do have quite a few questions and a couple of minutes to go ahead and answer those. So we'll go right through them here. First one: how long does it take for the insect growth regulator to absorb into the larvae's body and take effect?
I can't answer that, specifically. The biggest impact is in the later instar larvae, as I showed that the juvenile hormone levels decline as they age. Temperature is probably going to play a big role in that as well. The warmer it is, the more rapidly it probably could be absorbed. But that's a little bit out of my realm of expertise.
All right, well,
as many of these may be.
No, we'll go ahead and move on to the next one. Is there a mechanism of resistance to methoprene in mosquito larvae?
There has been examples of some resistance being documented, but there are many areas that have used methoprene for a very long time that do not have resistance. So that's kind of there seems to be some conflicting data on that. Chatham County, I talked to Bobby Mullis, and they've used methoprene for a very long time on their floodwater spoilage areas with great success. So the proper application, I think, is really critical to that.
Yeah, and speaks to what you mentioned about product rotation and the rest of that program of why that stays working well for them. So